Good day, everyone. I am Dr. Supache Basit, and welcome to another screencast in immunology and serology. And for today's lesson, we shall be discussing about the complement system. So, for this particular topic, our outline shall include the pathways of the complement system, the system controls, the complement receptors and their biological roles, the biological manifestations of complement activation, the complement and disease states, and the different complement deficiencies and its corresponding laboratory detection of the abnormalities. Okay, so let's define complement. So actually, the term complement was concocted by Pfeiffer and it literally means to complete. Okay, so what are we completing here? In complement, we are trying to complete several tasks, but here in the context of complement, with the ultimate output or goal would be cytolysis or cell lysis. So complement therefore is a series of more than 30 soluble and cell-bound proteins, and these proteins interact in order to enhance the host defense mechanisms against foreign cells such as bacteria. So, complement, incidentally, will also cause cell lysis of fragile cells such as RBC. So, most plasma complement proteins are actually being synthesized in the liver. So, for example, with the exceptions of C1, because C1 is actually produced by the intestinal epithelial cells, and factor D, because factor D is made in adipose tissue. So, WBCs are additional sources of early complement components such as C1, C2, C3, and C4. So, incidentally, C3 is considered to be as the most abundant complement component. Majority of the proteins, of these proteins involved in complement cascades are zymogens. So what are zymogens? Zymogens are actually inactive substances. Okay? So zymogens are inactive substances that are converted to enzymes when activated by another enzyme. Therefore, zymogens will actually be activated, okay, will be converted to activated enzymes in a very precise chronological order. Okay, so aside from cytolysis, some components of the complement may also serve as an opsonin. So opsonins are actually a substance that will mediate phagocytosis. So some complement components may act as opsonin, meaning specific receptors are present on phagocytic cells. And because of this, the metabolism and clearance of immune complex are enhanced. So when you say immune complexes, okay, these are made up of antigen, antibody, and complement. Okay, so complement may also increase vascular permeability. Okay, it will also recruit monocytes and neutrophils to the area of antigen concentration. Now, these particular monocytes and neutrophils are actually important in phagocytosis. So, complement may also trigger the secretion of immunoregulatory molecules that will eventually amplify the immune response. Okay, so in order for the complement to be activated, it will actually participate in three pathways. Okay, so classical pathway, we have the lectin pathway, and then we have the alternative pathway. So in some cases, there will be convergence of these different pathways, and that's the reason why multiple pathways can be involved as well. So as what I've told you before, the ultimate end goal or end product of complement would be cytolysis. So there would be lysis of the invading cell, such as the bacterial cell, and even in some cases of fragile cells, such as the RBCs. Okay, so remember that there are only two types of immunoglobulins that can activate complement, IgM and IgG. And for IgG, these are IgG1, IgG2, and IgG3. Remember, IgM is a pentamer. IgG is a monomer. Therefore, if we're going to compare the efficiency between IgM and IgG, IgM is actually more efficient. Being a pentamer, there are 10 FABs, okay, in IgM. So meaning there are multiple binding sites for the IgM. Okay, so let's talk about the classical pathway. Classical pathway is actually divided into 3 stages. So we have the recognition stage, the activation stage, and then the membrane attack complex. Okay, so the classical pathway may also be activated by substances that can bind the complement directly. C-reactive protein is one of them. So remember, C-reactive protein is an example of acute phase reactant. And then there are certain microorganisms such as viruses, mycoplasma, gram-negative bacteria, and even protozoans. So all of these microorganisms may activate the classical pathway. So let's discuss first C1. C1 is also known as the first recognition unit. Okay? Because it is the first complement component to bind. Okay, so let's contextualize the illustration. This is the cell membrane of the bacteria, bacterial cell. Okay, this is, for example, an IgG, then there's another IgG. Okay, so complement will not directly attach to the bacterial cell wall. Okay, it will not directly attach to the cell membrane of the bacteria. In fact, complement will bind at the Fc portion of the immunoglobulin. So this is the Fc portion. So what particular part of the complement? It is C1q. Why? Because C1 is consisting of 3 subunits. So in other texts, you will learn that C1 is otherwise known as the trimolecular complex, and this particular trimolecular complex is being held together by calcium ion. Okay? Now, that is the reason why if, for example, in serological assay, and you want to assay for complement, you cannot use anticoagulants such as EDTA. EDTA will eventually chelate calcium. Therefore, this will actually decay complement. Okay, so this is a trimolecular complex of C1. So C1 is made up of C1q, C1r, and C1s. So the one that binds at the Fc portion of the IgG is the C1q. So C1q has the capacity to recognize the Fc portion of 2 adjacent antibody molecules as shown here. Okay? 2 adjacent antibody molecules. So eventually, at least 2 of the globular heads of C1q must be bound to initiate the classical pathway. Why? How many globular heads, okay, does a C1q possess? So there are actually 6. 1, 2, 3, 4, 5, and 6. Now the requirement to initiate the activation of complement is out of the 6, you need at least 2 globular heads to bind with the Fc portion. Zymogens, okay, so remember zymogens? Zymogens are inactive enzymes that need to be activated, okay, by another active enzymes. And so once it is activated, then it becomes active enzymes. So there are 2 zymogens, the C1r and the C1s. Okay? So as soon as C1q binds to the Fc, C1r and C1s are activated. And at the same time, auto-activation of C1r results from a change that takes place as C1q is bound. So you have now here the activated C1r and C1s. Okay? So, when activated, C1r cleaves a thioester bond on C1s, which activates it. So, C1q binds, C1r is auto-activated, and C1r cleaves a thioester bond on C1s. So, C1s has a limited specificity. Okay, the substrate for C1s are the other two complement components, and these are C4 and C2. Okay, so when C1s is activated, the recognition stage ends. So again, as a review, okay, let's review, because discussion of the complement may seem complicated. So what will happen? C1q, two of the 6 globular heads of C1q binds with the Fc portion. This results to activation of C1r, and C1r auto-activates, and this also activates C1s, because C1r will cleave the thioester bond of the C1s, and C1s is activated, and that's the end of the recognition unit. Now remember, there are two possible substrates for C1s, and these are C4 and C2. Okay, so to animate, okay, this is the GIF animation. Okay, so by the way, the IgG binds at the epitope, and then these are the globular heads. You need only 2, and then C1r is activated. That's a zymogen, and that causes activation of C1s, and once C1s is activated, that's the end of the recognition unit, recognition stage. Okay, now let's discuss the activation unit. So remember, the activation unit has actually 3 components: C4, C2, and C3. Okay, C4 is known as the first activation unit. Okay, C2 is the second activation unit. C3 is the third activation unit. So C4, C2, and C3. Remember, in the recognition unit, you activated C1s. And what are the substrates of C1s? The substrates are C4 and C2. So what will happen is that C1s will cleave. So when you say cleave, in the parlance of biochemistry, you actually split molecule. Okay, so C1s cleaves C4 to create 2 components. Okay, so these are C4b, C4a, and the other one is C4b. Okay, and then C1s will also cleave C2. So this will result in 2 C2a and C2b. So C4a, as you can see here, will not anymore participate in complement cascade. C2b, as you can see here, will not anymore participate in the complement cascade. What is left is C4b2a. Okay? So the C4b2a is otherwise known as the C3 convertase. Okay? So that is otherwise known as the C3 convertase. So what is a C3 convertase? C3 convertase will cleave C3 into C3a and C3b. So so this is now your C3 convertase. So the C3 convertase will cleave so this is now your C3 convertase. It will cleave C3 into C3a and C3b. Okay? C3a will no longer participate in the complement cascade. What is left is C4B2A3B. The C4B2A3B is otherwise known as the C5 convertase. And what's the action of C5 convertase? The C5 convertase will cleave C5 into C5A and C5B. So, C5A shall no longer participate in the complement cascade, only the C5B. Okay? So, the activation of the C5 is the end of the activation unit. So again, in activation unit, the first activation unit is C4, the second activation unit is C2, the third activation unit is C3. Once C5B is activated, it is now the start of the membrane attack. Okay, so let's watch first the animation. So as you can see, once C1S is activated, it cleaves C4. C4A does not participate anymore, leaving C4B behind. And then it will also activate C2. C2B will no longer participate, leaving C2A behind. So C4B2A is also known as the C3 convertase. Okay? So the C3 convertase, which is the C4B2A, okay, so there are several C3, and C3 will be cleaved into C3A and C3B. So C3A will no longer participate. So there are several C3. It's the most abundant complement component. So what will be left behind would be C4B2A3B, and that is known as the C5 convertase. Okay, I told you a while ago that C4B2A3B is the C5 convertase because it will cleave C5 into C5A and C5B, leaving the C5B behind. C5A binds with histamine, with mast cell, and eventually it may cause vasodilation, and it attracts WBC, okay, because WBC may also have C3B receptor, and C3B receptor may actually will also serve as opsonin because C3B receptor here attracts neutrophils. Okay, and then some component of C5A will bind with mast cells, so they will actually participate in inflammatory and anaphylactic response. Okay, so some components of the C5B will attract C3B will attract neutrophil. So, okay, so you can see here, because there are several C3s, the most abundant complement component. So some component here will remain in the cell membrane, C3B. So what will happen? The WBC will undergo diakinesis, which means it will the muscle will allow the blood vessel to dilate and allows neutrophil to come inside, and there, chemotaxis of neutrophil. So that's one of the functions of C3B. So it mediates also in phagocytosis. Okay, so I told you a while ago that once C5B is activated, because C5A binds with mast cell, so what remains is C5B. Okay, so the activation of the C5B is the start of the membrane attack unit. So C5B is the first membrane attack unit, C6 is the second membrane attack unit, C7 is the third membrane attack unit, C8 and C9, these are the final membrane attack unit. Collectively, we call them MAC, okay, which stands for Membrane Attack Complex. So C5B attaches to the cell membrane to initiate the formation of MAC. So the MAC, therefore, is made up of C5B, 6, 7, 8, 9. So once MAC is formed, so there you have the C5B, and then it activates C6, and then it activates C7, and then it activates C8, and then it activates C9, final membrane attack unit. So there is now a hole and cell lysis occur. Okay, so that is the classical pathway. Okay, so another pathway of complement is the so-called lectin pathway. The lectin pathway activates complement by recognizing carbohydrates on microbial cell walls. Now, if you notice, bacteria, microbes such as bacteria, is made up of peptidoglycan. It's actually a mixture of proteins and carbohydrates. So, the lectin pathway may serve as a defense mechanism during infancy, because during infancy, there is, an awkward stage wherein you are losing the maternal antibody and the development of full antibody response. So during that transition, you may be vulnerable to infection. So fortunately, we have this lectin pathway to protect infant during this transition. So it involves 3 classes of recognition molecules. So unlike in classical pathway, it is induced by the IgG or IgM. This time, it is induced by recognition molecules such as lectins, picolines, CLL1. So what will happen is that after cleaving C4 and C2, then the process is now identical to classical pathway. So what's the difference? The difference is that the difference is that instead of C1, which is made up of a trimolecular complex of C1q, C1r, and C1s, in lectin pathway, the recognition units for molecules are lectins, picolines, and CLL1. And after that, C4, C2, C3, C5, 6, 7, 8, 9. So all of the cascade will be the same as the classical pathway. So the lectin pathway, therefore, may involve proteins, okay, such as the MBL or the Mannan-binding lectin, which is an acute phase protein. So guys, remember, when we're talking about the acute phase proteins, these are actually being produced in response to inflammation, okay? So the Mannan-binding lectin will bind to mannose or related sugar in a calcium-dependent manner to initiate pathway. So like your C1, okay, it is also calcium-dependent. And then aside from that, we have the so-called MASP, okay, the MBL-associated serine proteases. And there are actually 3 components or 3 kinds of this one, and they play an enzymatic role. So such as the zymogens that were just like the zymogens that were previously involved in our discussion of the classical pathway. Okay, so now let's talk about the alternative pathway. The alternative pathway acts as an amplification loop for classical or lectin pathway. So what do we mean by loop? Loop means it is happening all over and over again. That's what we meant by loop. So the alternative pathway can be activated by its own bacterial, fungal cell wall, viruses, tumor cells, or even some parasites. Okay, so what did I tell you about the most abundant complement component? C3, right? And in our previous discussion, you would see in the classical pathway, when C3 is produced, okay, so you hear C4, and then C4A, so C4B2A, and that is the 2A. So you have now the C3 convertase. C4B2A is the C3 convertase, and this will cleave C3. Okay, so as you can see, there are several C3 that will be activated. So C3 is the most abundant complement component. Now, why is it important? Because at this stage, the activation of several C3 is the initial start of the alternative pathway. So let's go back now to alternative pathway. Now, as you can see, you have here several C3b. Okay, because C3 has unstable thioester bond, it is constantly breaking down in plasma, wherein it is converted to C3b by ACP factors B and D. So we have now the ACP. So the ACP is simply known as the alternative complement pathway factors. And there are two ACP factors, and these are factor B and factor D. So C3 is constantly breaking down, and factor B and D cleave C3 all over again because it is constantly taking place. Therefore, it is otherwise known as the takeover activation. So it is the C3b that is attached to the microorganism cell membrane. You've seen the animation a while ago. If the C3b attaches to the cell membrane of the microorganism, it attracts neutrophils. So somehow, C3b may also serve as opsonin. But the story of alternative pathway doesn't end here. As you can see, once C3b is attached to the cell membrane of the microorganism, okay, so factor B is cleaved. So factor B is cleaved into two components. You have the BA and then you have the BB. So you now have the component known as the C3BBB. Okay? So the C3BBB complex, okay, is stabilized by a protein known as properdin. So that's the reason why in some other texts you would hear that alternative pathway is also known as the properdin pathway. So what will happen is that factor B, okay, is cleaved by factor D. So you now have the C3BB. Okay, the C3BB is also known as the C3 convertase. Okay, C3 convertase. And the C3 convertase in the alternative pathway. Because remember, in the classical pathway, our C3 convertase is C4B2A. But here in alternative pathway, our C3 convertase is C3BB. C3BBB. Okay. Don't forget, there are three B's. C3BBB. Siguro for us not to get confused whenever we pronounce big B, you have to pronounce it loudly. So what is the C3 convertase? It is C3BBB. Okay, there you have it. Okay, so what's the action of C3BBB? The action is, it is a continuous positive feedback. So it's always being produced, C3b is always being produced, and the C3b will cleave another C3. So if it cleaves another C3, then you have now an additional 3b, resulting into C3b B3b. Okay? So the C3b B3b is also known as the complex formula action of C3BBB on C3 is called C5 convertase. Wow! Okay, so for you not to get confused, in alternative pathway, C3 convertase is C3BBB, okay, which will lead into activation of another C3, it will cleave another C3 into, and then there would be formation of C3BB, B3B, and that is known as the C5 convertase. C5 convertase will then again cleave C5, and C5a, just like before, will not participate in complement cascade. So C5a here may bind with mast cell, okay, and then C5b, okay, will initiate the formation of C6, C7, C8, and C9. causing transmembrane pore because of the MAC, causing the lysis of the cell. So there. So what's the difference between the alternative pathway and the classical pathway? In alternative pathway, it doesn't start with C1. It doesn't need IgG. Okay? So it starts with C3. C3b in particular. Okay? There are components in alternative pathway that is not found in classical. Okay? These are factor B, factor D, and of course the properdin. And then there are components found in classical pathway that is not found in alternative. C1, C4, and C2. There's no such thing as C1, C4, and C2 in alternative pathway. See in our discussion, because it starts with C3. Okay, but again, the end result would be the same. So as you can see here, in the convergence of pathway, okay, so classical pathway, okay, will result into formation of C1, C4, C2. So you have now C4B2A, and then this is now the Lectin pathway is activated by MBL, or the MASP 1, 2, or 3 in the mannos of the microorganism, and then there would be activation of C2. Alternative pathway is activated by components of the bacterial cell wall, such as teichoic acid for gram-positive bacteria, zymosan and lipopolysaccharide for gram-negative bacteria, as you can see here. And then C3b is activated together with the factor B and D. Okay? So again, properdin is needed to stabilize the alternative pathway. But then again, all of these will converge into C5. And C6, C7, C8, C9, causing the formation of MAC and eventually lysis of the cell. So, sa hinaba-haba man ng prosesyon, darating din siya sa lysis of the cell. So, there are 3 pathways. So, it's like ways. The end result is the same. Okay, so there are several system controls in the plasma. So, for example, the C1 ion H is a C1 inhibitor with a molecular weight of 105, concentration of 240. This will dissociate C1R and C1S from C1P. So, this involves classical and lectin. Factor 1 will cleave C3b and C4b, affecting the classical and lectin. Factor H is a cofactor with factor I to inactivate C3b. Therefore, once C3b is inactivated, it can no longer bind with factor B to C3b. So it affects the alternative pathway. The C4BP acts as cofactor with factor I to inactivate C4b. So the one that is affected here are the classical and lectin. And then we also have the S protein or the vitronectin. It prevents the attachment of the membrane attack units such as the C5b, C6, C7 to the cell membrane. It affects all pathways, namely the classical, lectin, and alternative pathway. So these are, in a nutshell, this is how it works. Okay, how factor H affects a certain pathway. So there are also receptors in the complement. Okay, the cellular complement controls. The controls are actually being regulated by the presence of the receptor-ligand partnership. So receptor is found in the cell membrane, while ligand, these are substances that will bind with the receptor. So the receptor DAF, otherwise known as the CD55, the DAF or the decay accelerating factor, C3b and C4b will actually bind with DAF. Okay, so what are the cells that will have DAF? So this includes RBCs, neutrophils, platelets, monocytes, endothelial cells, fibroblasts, T cells, B cells, and epithelial cells. So DAF primarily function to dissociate C2b or factor B from binding site, thus prevent the formation of C3 convertase. So again, if there's no C3 convertase, C5 will not be cleaved. If C5 is not cleaved, then there's no cell lysis. And then we also have the MIRL, membrane inhibition of reactive lysis, MIRL, okay, otherwise known as the CD59. So the ligand or the one that will attach to CD59 is C8. So CD59 is found on the surface of RBCs, neutrophils, platelets, monocytes, endothelial cells, and epithelial cells. So if C8 binds with CD59, you'll not be able to insert C9 into cell membrane. Therefore, no cell lysis. And then the MCP or the CD46. The MCP is methyl accepting chemotaxis proteins. So the one that will bind with the CD46 are the C3b and C4b. So it is found in the surface of neutrophils, monocytes, macrophages, platelets, T cells, B cells, and endothelial cells. So it serves as the cofactor for factor I, which will cleave C3b and C4b. So if C3b and C4b is cleaved, you'll not be able to have C2 convertase. So that stops complement cascade. So we also have the biological roles of complement receptors. Okay, so complement receptors are found at the surface of the cell, and the one that will bind there, CR1 for example, or C3b, IC3b, and C4b. So found in these cells. So this serves as the cofactor for factor I, and it mediates the transport of immune complexes. Immune complexes are usually made up of antigen, antibody, and complement. Okay? CR2, also known as CD21, so the ligand or the one that will attach to it are the C3DG, C3d and IC3b. It is found in this cell. So the function is that it serves as the B cell co-receptor for antigen with CD19. Okay. CR3, so otherwise known as CD11 and CD18. So the ligands are IC3b and C3d, C3b. So the cell types that would have these receptors are the following, and its function is that it serves as an adhesion and it will increase the phagocytic activity of your phagocytes. And then the CR4, the ligands are IC3b and C3b and found in these cells, and it would have the same function as the CR2. Okay, so moving on, complements are very important because it will amplify inflammatory responses. It will facilitate recognition and destruction of antigen by phagocytic cell, and it has also a role in uptake and presentation of antigen, so a specific immune response can occur. So there are many roles, no? Inflammation, phagocytosis, immune response, and lastly, B cell activation. B cell activation is very important so that once B cell is activated, it will be differentiated into plasma cells, and plasma cells will be the one to produce antibodies. Okay, so complement is associated with both natural and adaptive immunity. As what I've shown you before, it amplifies inflammatory response. Remember, C5a binds with mast cell, so C5a may serve as anaphylatoxin. C3b will serve as chemotaxin and opsonins at the same time. So there are other important responses or components of complement or important roles of complement in our immune system. However, complement can be harmful if it is activated systematically on a large scale. So for example, you have gram-negative septicemia, meaning bacteria are found in your blood. So again, the drastic immune response may not be beneficial to the host. it is activated by tissue necrosis. So, as in myocardial infarction, that is also dangerous. Okay? Or if lysis of RBC occur, because that will result into hemolytic anemia. Okay? So, these are conditions wherein complement can be harmful. So, complement is important for our immune system. Meaning, if you are deficient for a particular complement component, these are the associated disease. Imagine, if you do not have C5 or C8, Neisseria infection will occur. And again, what is the most abundant complement component? C3. So deficiency of C3 may result to severe recurrent infections. So can we measure complement in the lab? Yes, it can be measured. So measurement of component as antigen in serum, that's possible. Our measurement of functional activity, that's also possible. In fact, in the latter chapters, we will be discussing the complement fixation test. Although complement fixation test, complements there will actually will bind either with the indicator system or the antigen antibody complex. But that's another story. Or the most common way of measuring complement component would be by means of RID and also nephelometry. So classical pathways can be measured by measuring the ability to cause lysis, such as the CH50 assay. CH50 test for lysis of liposomes and the radial hemolysis in agarose plates. For alternative pathway, we can measure it using the AH50 and ELISA. Okay, so there are even several test systems addressing all three pathways. In fact, we can interpret laboratory findings. Okay, so for example, if C1q, C1r, C1s are low, meaning only the classical pathway is affected. Why? Because these C1 components are not found in lectin alternative pathway. So for example, if factor B, D, and P are low, only the alternative pathway is affected because these factors are not found neither in classical nor in lectin pathway. Okay? So if, for example, MBL and MAST2 is low, the only lectin pathway will be affected because MBL, MAST are not found in classical or alternative pathway. So if I were you, please study this. Okay? Study this table. So as you can see, if C5, for example, is low, C3 is low, all of the pathways are affected. Okay? So be sure that you'll be able to study this table. So there you have it. You may want to screenshot the summary. Okay, so that's the end of our discussion on the complement cascade. Complement is actually one of the first time I have actually read this in immunology book, so I find it difficult to understand. So I hope that you're able to learn something about complement for today. So God bless everyone and stay safe. See you soon.